Welcome to Hack Your HazCom. Today, my guest is Rob Mitkus, who has worked as a toxicologist for the past 25 years outside of graduate school, where he earned a PhD in toxicology from the University of Maryland at Baltimore. His career in toxicology and risk assessment has spanned multiple sectors, including industry, government, and consulting, as well as classes of agent, including traditional and non-traditional pesticides, industrial chemicals, vaccine components, medical devices, and consumer products. He's certified as a toxicologist by the American Board of Toxicology and is a European registered toxicologist, an SDS RP. He currently works at ITG Brands, and he has also recently completed a master's in industrial and organizational psychology from St. Leo University. Welcome today, Rob. Thanks for coming on.
Thank you, Atanu. It's a pleasure to be here with you.
Yeah, great. So thank you for being on today. And what I wanted you here for is a perspective from the toxicologist point of view. In what we do all the time here with our safety data sheet authoring, our HazCom compliance, it's very important to have knowledge of toxicity, toxicology, and what better person to have on today than you to delve into some of this. So, let's talk about it. What is toxicity and how does it relate to toxicology? And why should we care about it?
Sure. Yeah, these are great questions. And I wrestle with these questions every day in different contexts. So essentially, toxicity is harm, or adverse effects, adverse events. There are different ways of, phrasing it, depending on the paradigm you're in. But essentially, we're concerned about, in toxicology, we're concerned about reducing the risk of a particular adverse effect or harm to humans and other species, essentially. And so ideally, toxicology is meant to be a discipline that examines those effects and the conditions under which those effects may or may not occur. There's a little bit more to it than that too, but essentially that's what toxicity is, harm or adverse effects to an organism under certain conditions. At least that's how I think of it.
No, great explanation. Yeah. So
Yeah.
in what we do with safety data sheet authoring, it's up to us to communicate something of a hazard and what it is to the worker and ultimately worker safety. Why is toxicology important with that?
Yeah. So toxicology, the study of these adverse effects and the conditions under which they occur, it forms the bedrock and information, ideally, and evidence that should go into an SDS. So, I mean, as you know, Tanu, SDSs contain a whole section just on toxicity and toxicology effects in humans primarily. And then we also have another section for ecological effects. But ideally, SDSs and those sections specifically, and then the precautions that we would recommend in an SDS should really be driven by evidence of adverse effects. So in toxicology, whether we study these effects in animals or we use in vitro methods, we're trying to develop evidence and data that can then inform the content of SDSs. So it really wouldn't serve either the public or stakeholders that utilize the SDSs to make recommendations or classifications without evidence. So that's why why toxicology is so important to SDSs.
Okay. And you touched upon this with stakeholders. Ultimately, what OSHA wants us to do in this with the safety data sheet is consider all the downstream users of this document. First and foremost is the worker that's actually handling the chemical right then and there. But of course, we have these other stakeholders that we need to address. How do we think about toxicity for these stakeholders? And if so, who are these other stakeholders?
Yeah. And so, yeah, exactly. That's exactly right. The end user of the SDS we have to consider, as well as potential population so that may be exposed to whatever compounds or mixtures that we're writing an SDS for. So as you mentioned, first and foremost, or primarily workers benefit and can use SDS information and SDSs themselves. Consumers as well are another downstream user of SDSs. So whether that's, you go to the Home Depot and you buy a pesticide, usually they don't have an SDS associated with it, but you can request one from the manufacturer. And so, sometimes for consumers, a safety data sheet may be all you have in terms of assessing, what should I be concerned about? And we'll come back to this, I think, later on too, but this idea of what should I be concerned about is related to this idea of risk as well as toxicity. So yes, the GHS that forms the basis for SDSs talks about the end user and downstream users of SDS, the target audiences. That includes workers, consumers, first responders, and others. But so the SDSs, this raises the question of when SDSs are, developed and written, not only should they be based on evidence, so evidence of toxicity, but also they need to be comprehensible for those downstream users. So
communication piece, right? Exactly. Who is your audience, right?
Exactly. And I think we'll probably come back to this too, soon at some point. The idea SDSs, are we, for that hazard communication and risk communication piece, are we sometimes over communicating with the SDSs, under communicating, or do we, get it just right, like Goldilocks? So
sure. Great point there. And I think you mentioned this earlier about where we get into like ecological aspects, environmental pieces, right? So a lot of the safety data comes from, non-human species, right? Invertebrates, fish, birds. Should we really care about them? And as far as what does that have to do with what we need to rely upon with this data?
Yeah, right. Exactly. Yes. So yes, we should care about other species. And this idea of different species has, I think, two components. So the first aspect gets back to where we kicked off, our discussion about toxicity. And so, many cases we don't have human data, that really robust human data on which we can use to write our SDSs that informs the toxicity. So historically we relied on animal studies. And so there's that first piece where animals, other species, primarily rodents, there are other species too, but in the chemical space, largely rodents, have served as what's called sentinel species for humans. So, the SDS is, we're trying to protect workers from potential exposures, but not just workers, but for humans that we're interested in protecting, or at least raising communication, we rely on non-human species, sentinel species, to help inform the toxicity and toxicology. The other aspect of SDSs, as you're implying, is, there are other species besides humans that we're concerned about in an SDS. And so, and the GHS goes into this in depth, so we have human health hazards and then the ecological health hazards. And so I think we are concerned for various reasons of potential exposure to non-human environmental species that could be exposed to a compound or mixture for which we've written an SDS. Now, GHS, as you know, it focuses primarily on aquatic hazard, but we're not just interested or concerned about fish and other aquatic species. We are concerned about other birds, and so it raises the question, do we need a section on birds in the GHS? Maybe, maybe not. Perhaps, the idea of having aquatic, toxicology there, or aquatic hazard as an emphasis, is in case there's a, runoff into lake, other bodies of water. Whereas if, say, there's a an accident, a spill of some sort, and it's out in the open, then the old, dilution is the solution to pollution,
Right.
maxim would apply. And so might be less of a concern, but yes. So, and GHS kind of spells that out. The GHS document spells that out of human species and then non-human animal species.
Now, this concept of sentinel species, what is the criteria for selection of that?
Yeah. So it's probably driven largely by historical practice. So, I mean, we could go back into the history, FDA in the 1940s set up an early risk assessment paradigm for drugs and evaluating drugs and risk assessment for drugs. And those relied at that time, we're talking about 1940s, 1950s, on rodent species and canine species. Over time, those species have kind of been just part and parcel of the toxicology paradigm in terms of experimentation and using those whole animals as sentinel species, number one, to generate data on adverse effects, and then number two, to extrapolate from those data to humans as part of a risk assessment framework. Over the last, few decades, we're moving perhaps beyond that paradigm. I think there's a lot of calls from different sectors to move beyond the animal testing paradigm into we'll get to this, I guess, new approach methodologies. But at the same time, in drug development, at this point, there will be no testing of a drug under development in humans without the non-clinical species testing. So the sentinel species testing. Yeah.
Great. And that dovetails nicely. The alternative to animal testing, what are they and how does it relate to our chemical classifications that we do in the GHS system?
Yeah. Yeah. So these new approach methodologies or alternative methods are very hot right now. It's started to get hotter, I think, over the last 5 to 10 years. And there's a lot of effort from various organizations to validate alternative methods of assessing toxicity. So without bringing a compound and testing it in a whole animal, and even less so in humans. So moving into, in vitro methods, in silico methods, predictions. But essentially a new approach methodology is any approach that doesn't utilize a whole organism to determine or measure toxicity, and then from there extrapolate to risk. So, we've had this it's been, in terms of experimental toxicology, in vitro methods have been around for decades, and kind of the movement now is to see if there are in vitro human models, cell models, whether that's organ on a chip or other in vitro methods that can be used to predict toxicity in humans. And I think that's the most important thing. How predictive are the methods we use now in predicting toxicity and risk for humans or other species? It's that predictivity piece that's the big question.
Yeah. With that, and I would imagine these novel approaches, new approaches, do they get vetted, or who agrees? Is it by consensus? Who are the bodies that come up with the, a consensual based approved NAMs?
Yeah. So very important question. And so essentially, yes, there are certain bodies out there. So there's OECD, Organization for Economic Cooperation and Development. That body, they have certain committees that validate assays, both in vivo but also in vitro, in these new approach methodologies. ICCVAM is another international organization that brings together experts in toxicology and specific methodologies to validate these new methods. Even within certain regulatory organizations, for example, FDA, they have committees internally and also policies and guidance when it comes to these new methodologies. More on the side of uptake and regulatory acceptance, what they would accept and not accept. But yes, it's essentially a consensus. I guess you can consider it a consensus set process where organizations, committees within OECD, ICCVAM, ICCVAM, I think is another organization that NTP has, where these methods are examined, sensitivity, specificity, different validation criteria need to be met before they can be really used, in prime time for making predictions for humans, and then being submitted to regulatory agencies. Yeah.
And are these widely accepted, like not just here in the US? Are we talking, worldwide? What is the general acceptance of the NAMs?
Yeah, so it varies. It varies. It varies by region. It varies by regulatory or organization. So this is why. So OECD, I would say, in terms of if you wanted to, again, across region, if you wanted to use as a resource what methods have been validated in terms of NAMs and in vivo, OECD, I think, is a very, very transparent and open organization. You can go to the website and all of their test guidelines can be downloaded. You can, sort just on specific NAMs. There aren't that many yet, historically, that you would still have a lot of in vivo tox guidelines because in vivo tox studies are, conducted routinely. But for example, skin sensitization, there's a whole NAM for that. And I won't get into too much into the details there, but there's 4 key events, and each of those key events can be assessed by a different in vitro or, again, I'm going to get into the Latin, in chemico. This is the new in chemico method, whatever your favorite,
Medium.
Latin medium. Yeah, exactly. And that's been validated. That's
Okay.
a validated assay, or called a defined approach to skin sensitization. Unfortunately, some of the more complicated endpoints, like carcinogenicity, reproductive toxicity, they're a little harder to boil down into quick, or less complicated in vitro assays.
Okay.
So I think, going forward, there is momentum towards and in the NAM space, even for in vitro lung models to assess local toxicity from irritants or other xenobiotics that we might inhale. But I think, there's over time, looking into the future, there's still going to be difficulty in, boiling down or using in vitro models to mimic some of these more complicated organ systems. Toxicity seen there. So, but yeah, skin sensitization is a very good example. Another example we've been using, it's not a NAM, but we've been assessing genotoxicity in vitro methods for decades, going back.
Okay.
So not a NAM, but in that space, genotox, the NAM would be, new approach methodology would be these in silico, we're called in silico methods, computer models, QSAR, making predictions about potential genotoxicity or carcinogenicity. I'll come back to that later, but yeah. So yeah, does that give us somewhat of a flavor?
Yeah, it does. And I think that skin sensitization example, has that been used, I think, by the EPA? Was that in some time recently, or could you talk on that?
Yeah, sure. Yeah. So, about 6 years ago, I became aware of actually EPA using this, Office of Pesticide Programs, use this defined approach in assessing skin sensitization for isothiazolinones as a class of dermal sensitizers. And so, yeah, what they did there was they had a mixture of these in vitro assays, and then they combined that with what's called a machine learning approach, which is really using computers and complex statistical programs to make predictions about, the robustness and reliability of the results. And so they took that approach for that class of compound to make predictions about dermal sensitization for risk assessment. So that was in terms of how the defined approach and what is kind of like AI, the machine learning. Yeah, it does. I believe it does fall under that moniker, but now AI, the last 5 years, gotten really, really hot. But yeah, that was kind of one of the early times that I've seen that combination of a defined approach as a NAM, along with an AI approach as a NAM, brought together to make predictions for humans.
Fantastic.
Yeah, there are other examples, I'm sure, but yeah,
Sure.
that's just one that I'm familiar with.
Okay. Yeah. And going back to, how complicated a human model is, like, how do you make that jump from, statistical data to what actually goes on in a human body, right?
Yeah.
And I would hope AI is helping with that, or they're working on ways of doing something that's more reliable that way.
Yes, that's the aspiration. That's the aspiration. And I'm sure, as you've seen, I mean, just for AI more broadly in the market, the question, I mean, it's an open question. You have CEOs of various companies, marketing and trying to sell AI. And there are a lot of people who have bought into that. But how predictive it is and what it can do, I think is still an open question. And that's the case, not just for, let's say, in the military, that's for the battlefield where it's, that's a headline we've seen recently,
Right.
But in terms of toxicity as well, making toxicity predictions, how well can AI be used to make predictions, let's say in the drug development space, or drug and a target, receptor or some other type of target to make predictions. I think we'll have to wait and see. I can tell you, outside of that, like for chemical risk assessment, it's not used routinely, in most settings.
Okay.
The AI, but does it hold promise? I think so. It's real again. It's like any system that humans have developed and that humans, set the conditions for and then inform. So
Right.
we have to give, a system all the information it needs, and then to make predictions. And we've seen how poorly or well it's worked with ChatGPT. Even academic programs are using AI. Certain programs turn it in as one program to look, students send in a paper, and then they're looking, was this taken from somewhere else? What's the probability of that? So don't want to get too much off topic, but yeah, AI, we'll have to, I think it's a wait and see. That's my view.
Yeah. I think, like most things, let's see where it goes, but have the human behind the wheel, right? That's the key. Let's hope.
Exactly. And, whether it's an AI model or an in vitro model, we're trying to make predictions for an in vivo situation, whole organism, then we have to be concerned about false positives or false negatives. Historically, regulatory agencies have been, and I think it's a true bias. They're biased towards concerned more about false negatives, whether that's a balanced approach or not, we can debate. But, I think, if we're talking about Goldilocks, we want to be in a situation where the bed is not too soft nor too hard, but just right. We need to be concerned about both false negatives and false positives. And so AI, in vitro, the NAMs, those are questions about those approaches.
Sure.
Just like in vivo studies.
Yeah. which we've had for ages, right?
Exactly.
Eons.
Eons, yeah.
So great, great. And, jumping back a little bit of a deeper dive with the stakeholders of the SDS, right? We talk about how we're communicating. Is it being comprehended by the right audience? Do we think we're communicating the same information to these stakeholders or the end users? Should it be, achieving this, or are we falling short?
Yeah, so I kind of have a view on this, just the way SDSs are created. But I have to remind myself like why SDSs are there to begin with.
Mhm.
So, SDSs are basically hazard-based classification. I mean, there is a section on risk to consumers. But primarily the focus is to present potential hazards for a given substance or a mixture. And so, the question becomes, someone who has an SDS and they're reading it, how are they reading that? What's going on in their mind? And I think that's a question I always ask. And I think it's probably different based on which stakeholder you are.
Right.
So, like a worker in a manufacturing plant is probably asking himself, if I'm going to be exposed to this chemical or use this chemical, am I at risk for fill in the blank? Some physical hazard, some human health hazard. A consumer, someone who's at home, not in a manufacturing situation where they may be exposed to a chemical or a compound day in, day out, I would say probably thinks about SDS a little bit differently. But even then, I have to step back and say, even across, consumers and workers, people have different risk tolerances, tolerance for risk. And not only that, but levels of understanding of an SDS. So this gets back to this, idea of what is the SDS communicating? So, we have pictograms, and I'll just take, for example, skull and crossbones. Like when you see a skull and crossbones, what's going through your mind when you see that?
Right.
And so, for me, I would see that and say, okay, this is deadly? Is that what that's trying to communicate to me? But, when you go beyond the surface, and I think this is important to do, and you maybe dig into section 11, go into the actual data that hopefully is there for on an SDS, and you get a little more information, maybe you won't be as concerned. So this gets to that question, are SDSs over communicating or under communicating? Do they get it just right? I think pictograms in general, you have a fish floating upside down with a dead tree, no branches, or a human with, it looks like
chest. Right.
The exploding chest looks like Iron Man, Iron Man with, all the energy going in there, but energy's going that way. So yeah, those are really, I would say, dramatic. Skull and crossbones, exploding man, dramatic, a dramatic way to communicate hazard or risk. And so perhaps in certain regions of the world or conditions, maybe where there's no regulatory apparatus, infrastructure, where compounds could be used with very little PPE, let's say we're out in the field, but compounds or mixtures are handled without any PPE. Perhaps psychologically, that's a way to make someone stop and say, okay, I need to take a closer look here.
Sure.
But, and the cross stakeholder too. So even, that we assume that. But yeah, I think in terms of where toxicologists can maybe help with that, hopefully, is to kind of invite and encourage readers of SDSs, certainly in companies, to take the next step beyond just the symbol and then the hazard classification, and then ask the question, okay, you know, what are the exposure, if I'm going to be exposed to this compound in the, say, as a worker, what are the levels I may or may not be exposed to? And then what is the toxicity I should really be concerned about? So I come back to this in a different way too, soon, talking maybe about the difference between toxicity and risk, and kind of what the GHS gets right and what it doesn't
Yeah.
get right, in my
I
view.
love that. Yeah, absolutely. So, and similarly, we all have our marketing groups, right? That tend to push back. That going back to that skull and crossbones. Oh my God, if we put this out in the market with that symbol, how are we
Yeah.
ever going to sell? What's the pushback there? And what is your advice on dealing with that?
Yeah. I've dealt with that in the past. That where a particular formulation was evaluated under in one set of conditions, and it was then evaluated under a new set of conditions, and we got a category 1 inhalation risk for it. And then the question was, okay, how do we communicate this to the business? And so, I mean, my recommendation is you go with as a toxicologist, as a compliance person. Those are different functions, but if you're working, in compliance or SDS authoring and creation, is go with the evidence.
Mm.
That's my basic recommendation. So if credible evidence shows, yes, I mean, okay, we have these tox results. This is where it puts us in terms of the GHS classification. I mean, yes, present that to the business side, and you just be transparent and open and say, here's what it is. Am I concerned really from an end user perspective, downstream end user perspective? Probably not. If this would be part of the communication to marketing as well. Probably not. If the PPE is followed, or the product is used as it should be used. But yeah, so those kind of conflicts arise all the time in businesses, and parts of fiefdoms. Sometimes they're fiefdoms within an
Sure.
organization. And I think just my advice is be transparent. Communicate what the data show, what the evidence shows. Doesn't mean it's going to kill your product. It may lead to some risk in the market, as you mentioned, but I mean, what are the alternatives? You have to, you can reformulate.
Right.
I guess that is one.
Sure. It
But
gets you
yeah,
thinking, right? Yeah, it gets you
yeah,
thinking, what are
exactly.
my steps? Yeah.
What are your steps? Yeah.
Yeah. Because I think, going back to that Home Depot example, right? It's your consumer, the home consumer that needs to work with something quick basis, but then it happens to be, I don't know, methylene chloride with some nasty thing in it. What do you do there? And now they have this SDS. Do they understand what that means, right? And how does Home Depot communicate that hazard to the end user? So you've got all these downstream cases where in Europe, that's known. You have to tell your consumer this. Where's the balance?
Yeah, that's a good question. Yeah, and there are definitely differences across the pond between Europe and the US. There's just also, I mean, like, say we're talking about a pesticide or,
Mhm.
let's say any chemical you might use around your garden. A lot of times there's no SDS on the label, but you open it up and it's like 20 pages that flip out into like an accordion, and and that just, I think that overwhelms most people.
Oh, yeah.
Probably going to get some minority of people who are interested and will take the time to read that. And so then the question is, well, how do you communicate hazard and risk, potential for hazard, accurately without alarming people? And so yes, I don't have an answer to that one, Atanu. I think other
We've
than
been
taking
dealing with that in for ages too, but your perspective makes some sense as well. So important to know, right?
yeah, it's important to know that there are those differences for sure. Um, reading the label is probably foundational. Yeah. Reading that label. Um, and I think most consumers aren't going to go beyond that if they even read the label. But read the label and then, precautions, what should I be, preventive measures. Should I wear gloves with this or should I not? Do I need eye protection? I think those are probably key pieces. Because, just looking at a skull and crossbones or even an exclamation mark, I mean,
What does that mean, right?
what does that mean? What does that really mean? We always should be going beyond that, but that's why, so that's why I'm kind of on the fence about how well SDSs or GHS communicates risk. And to the extent that things are harmonized, I think you are not harmonized.
No.
They're not. So
Right.
GHS.
I always say generally harmonized, right? So
Right.
what does that
Right,
even
not
mean
globally.
across the globally? Right, exactly. And
Right.
related to that is these classifications that we're using. Ultimately the GHS gives us these rule books to follow, right? The classes, the labels, are these actually accurate? And do you have examples where it's probably incorrect? And what should we do about that?
Yeah, I've thought about this, having, gone through the GHS quite a bit and just, and non-GHS doing risk assessment for so many years. So I think what the GHS document gets right is its approach to try to, look at different types of toxicity. So here I'm talking about human health hazards.
Mm-hmm.
So repro, carcinogenicity, and down the road, acute tox. Even some of the categories within a particular class, are helpful. For example, eye irritation, a 1 versus a 2A or a 2B and that type of thing can be helpful. Then again, you're always, those classifications they're, let's say one, 2, or 3. Let's say you have three categories. Ideally, you end up in each one of those categories based on evidence, science, evidence, which is not just 3. It's a continuum, much broader continuum,
Sure.
Of evidence. So to the extent that you collapse, tox data down into 1 or 2 or 3 categories, you're always losing information when you do that.
Right.
But to the extent that GHS is attempting to classify mixtures or substances based on toxicity, I think that's a good thing. Where I think GHS falls short, and there's one major way and maybe a smaller way, where I think it really needs to be revised is the document refers to what intrinsic hazard or intrinsic toxicity. And that phrase it's really not accurate. It implies that, and it actually states, GHS states, every compound or every substance or mixture has an intrinsic toxicity. Well, toxicity is not an intrinsic property. Yeah, like atomic number.
Right.
Or others. Toxicity depends on many factors. So, species, route of administration, length of administration, how the compound is formulated. So, for example, chromium-6 versus chromium-3.
Mm-hmm.
You get still the same element, chromium, but chromium-6 is a known carcinogen, operates through a mutagenic mode of action. Chromium-3 is a dietary constituent. So we need chromium.
Functions. Right.
So the oxidation state is chromium-3 versus chromium-6. So is chromium intrinsically hazardous? Well, no, it depends.
Right. It's too broad of a brush, right? Is that what we're saying?
Exactly. And then the problem, so it's riddled. The document is riddled with that phrase, intrinsic hazard, intrinsic toxicity. It gives the impression that the dose doesn't make the poison. We know the dose does make the poison.
Which goes against toxicity in the first place. This toxicology, the study of, right?
Exactly. Right. Exactly. It contradicts the basic tenet of toxicology that is actually it's an accurate statement.
Inaccurate.
So that's the one thing I think. But again, the document's announced in 11th revision, and it gets carried through each time. And to the extent that people are interested in toxicology or not, but are using the GHS document to learn about toxicology or hazard, but have no other resources, they're going to go away with that idea. And it's not an accurate, that there's intrinsic toxicity.
Maybe we need more toxicologists when we do updates to the UN rules, right? In the mix.
Yeah, I think so. Yeah. Because what I've seen in my career too, is that phrase then just gets uncritically carried over into other contexts. So I worked in medical devices for a few years in consulting there. I had colleagues across the pond who were taking that phrase and putting it into risk assessments. And so it's just not accurate. And so, yeah, we want to be accurate, in our risk assessments and risk communication, and hazard communication.
Interesting.
Yeah.
Go
ahead. Go ahead.
Yeah, no, just as an aside, I mean, going back to now AI and learning models, you've now introduced that concept as a bias potentially, right? If that's used across the board, what does that mean for risk-based stuff using AI?
Yeah, that's right. Yeah, right. If you have an AI model you're developing and you give it a training information off, it's going to train and then make predictions and spit out predictions. Yeah. If the information just going into it is incorrect, what's going to come out the other side is going to be just as incorrect. So,
right.
Yeah.
No, I'll have you on a future episode. We can get into some detail there, because that's going to be the future of what we're doing with this stuff, with the risk-based modeling.
Yeah. Yeah, absolutely. Yeah. I mean, and, to the extent that AI can help with the risk side, toxicity and toxicology are already kind of context dependent. And so, yeah, it's not going to be a situation where it's going to be 1 + 1 = 2. And yeah.
Shades of that. Right.
Yeah. Yeah.
Interesting. So, and I think similarly, the definition of sensitization, do we get that wrong?
So in my opinion, I think, yeah, GHS gets that wrong. And I've seen that carried over in other contexts as well. Or, I was attending a CE course maybe 6 months ago online, and the person was talking about dermal sensitization and risk by the dermal route. And lo and behold, they're using, the definition from the GHS. So GHS defines either respiratory or a dermal sensitization. So in the definition, they say it's either an allergic response or hypersensitivity, and that's not correct. So we're concerned about that applicable outcome, say dermal hypersensitivity or allergic reaction. That's what we're concerned about in the organism. But sensitization is just one step in that process. It's the initial step. So sensitization or induction. So you need a previous exposure in which you're priming the immune system to then react, because I won't get into the details, but they're calling in memory T cells in that second step. It's called elicitation. So, and then that's your hypersensitivity reaction. So poison ivy, for example, you're out in the garden. And by the way, I think 10% of humans do not develop allergic reaction to poison ivy. Most do, but yeah, there's about 10% that are immune from it. So the 90% who do develop it, it's this immune mediating reaction. It's not just irritation. So you need an initial exposure, which there's no clinical outcome for that. There's no rash. There's nothing you can see or measure visibly. So that's the priming or the sensitization step where you're exposed initially. Below the surface of your skin, your keratinocytes, your dendritic cells are doing their thing. Your immune system is, protein from the poison ivy or the oil is being presented, to the immune system. And then there's an immune development, but it's not until you get exposed a second or third time subsequently that you actually get a rash. That's what I'm trying to say. So sensitization refers just to that first step. And elicitation is the second step. But in the GHS, sensitization is being used to refer to the entire process, both steps.
Yeah.
Yeah, as a shortcut. Exactly. That's what it's being. But then it's not correct, biologically. And so that's an easier fix, I think, in future, versions of it. I'll just redefine it. The outcome we're concerned about is, hypersensitivity or allergic reaction. And then the downstream effect of that too, Atanu, is there are certain assays which actually do measure sensitization, like the local lymph node assay, that is focused just on the sensitization/induction phase. Whereas other, like, the guinea pig assays, the Bueller test, that actually, excuse me, you're assessing both steps, sensitization and elicitation. So Bueller actually is the more relevant assay, but it's actually assessing more than sensitization.
I see.
So,
encompassing more in the test.
Yeah, it would be a more relevant test in terms of predicting actual allergic reaction or hypersensitivity as an outcome.
See, so this is more reason where the audience, I hope, if you're listening, the UN people out there, we need more toxicologists as part of the process, right? In future versions, correcting things. It sounds like we need more of that from what you're saying.
Think so. Yeah, exactly. And again, I don't know who's on the committees or boards who are revising this. And, but yeah, I definitely would say yes. Get some individuals there who are aware, know this basic biology, and can inform that. Because I think I understand what they want to protect against. You want to protect against that initial sensitization step, but it doesn't help to define the entire process inaccurate.
Okay. Other than what we've spoke to earlier, do you find any other recommendations that we could use to maybe improve on the concepts of communication and hazard recognition, perception out there?
Yeah, yes. And this is one which I'm dealing with regularly. And this is the idea of carcinogenicity and genotoxicity, the risk of developing a cancer of some sort. So, and this does link up to GHS, and I'll explain in as well. But so even more broadly beyond GHS, regulatory agencies tend to, many do, not all, some get it, but many tend to classify compounds into a carcinogen or non-carcinogen, genotoxic or non-genotoxic. And that binary kind of classification isn't helpful at all. It just doesn't reflect, kind of like, how intrinsic hazard doesn't reflect the real world.
Sure. Similarly,
these binary classifications also are not helpful. And so this connects directly with, SDSs and GHS, is that classifications, let's say CMR, right? Carcinogenic, mutagenic, reproductive, toxicant. Classifying a compound into any of those categories is not helpful because it doesn't give you any information about the condition under which the carcinogenicity was observed or reproductive toxicity was observed. There's no dose information.
Okay.
So this is something which I think is kind of endemic into all hazard-based classification systems. And SDSs and GHS is a hazard-based classification system, not risk-based.
Right.
And there are hazard-based classifications for example, in Europe, CMR classifications, CLP classifications, which are hazard-based. And it gives the impression that I should be concerned about exposure, any exposure to a compound, a compound at any exposure,
right,
when it's regardless whether if it's, labeled as a carcinogen or a genotoxicant. And I wish I had time because I don't want to get too much into the weeds, but yeah, we could talk about like genotoxicity studies too, the in vitro ones, and, whether it makes any sense to label a compound as toxic when it was only seen at like the highest concentration tested and all the other lower concentrations were negative. Again, the dose does make the poison. So binary classifications, like that's a carcinogen or not, that's a mutagen or not, just it's an outdated way of thinking about chemicals, about toxicology. And there's another one I would just add to it real quick,
sure,
which is this low dose linear in terms of carcinogenicity, drawing a straight line back to zero, about cancer risk. So as you know, for carcinogenicity studies that are conducted in animals, many doses are tested. In most cases, you see, if you do see tumors, there's going to be a threshold. So a dose or concentration at which you see no tumors. This risk assessment approach is of ignoring the shape of the dose response curve and just drawing a straight line back from that lowest dose as it, again, outdated thinking. That's the type of thinking and understanding that existed at the time of the Delaney Clause in 1958, which the FD&C said, any compound that has shown carcinogenicity should be banned. So thank goodness, our understanding.
we've evolved.
We've evolved, yes. Amazingly, beyond that. But this low dose linear approach is like a default approach used by, and I won't name them, but several regulatory agencies still use that. And it's just not accurate for the majority of compounds that have been studied. But it's easy to do, you just roll. Yeah. And we reassure ourselves that we're, being protective and doing something, but it's not. This is where that difference between, toxicology and having a good understanding of toxicology differs from just, following a rote default approach to things, or, doing things because we want to comply with, a regulatory statute. That difference, this highlights that as well. So all that's really saying, what I'm trying to say there, Atanu, is that, it's really important to step beyond the hazard-based classification system, and look beyond that. If you really want to have a good understanding of what a compound is or isn't doing, should I be concerned or not, you really need that exposure piece. You really need that dose response piece. You really need to look at the underlying data. And that a good toxicologist will want to do that and will recommend that, as opposed to just going with the, default assumptions. So, yeah.
Great advice there. I think, again, very important to have somebody knowledgeable and able and willing to communicate to the user, right? And ultimately that goes back, going back to where we are with SDSs and, why we do this, the comprehension, communication of this. Do you feel like we may have a psychology there, or like an understanding that we need to have in mind when we put these things out there?
Absolutely. Absolutely. So, and that ties back to the skull and crossbones idea. So, and this isn't really touched on. I mean, I've been to a lot of CE over the years. I've been on many projects over the years, and the psychology of this is not really touched on. So, I mean, I got a master's in psychology as a side gig. And so I would, because I'm interested in that, but absolutely. So I would just boil it down to this. So it has to do with threat detection in terms for human beings. you see a skull and cross, we're already wired to be sensitive and to look for threats in our environment as mammals. That's us. That's how we're wired, as animals. And so our amygdala, is very easy to fire. But we also have this thing, it exists behind this, massive bone here called the prefrontal cortex, which has inputs into the amygdala, along with the thalamus and other sections. So we're not like reptiles where we just react, but we do have cognitive inputs into how we react to things. Now those break down under certain conditions, but because of that, when we perceive a threat, you can have an automatic reaction to it, like seeing a skull and crossbones. My immediate reaction is, well, that's bad. Like that's a skull and a crossbone. That implies death, right? That's bad. I'm going to avoid that. But then, so that's kicking in the fear response and the fear machinery in the neurological pathways in my brain that are trying to detect threat and keep me safe from threat. But because I have this prefrontal cortex, that can also kick in, and I can remind myself, okay, wait a minute now. Okay, yeah, that's this, that's the symbol. But maybe let's see what and it has a signal word associated with it. Okay, but let's go to the next step, talking about the SDS. What's the hazard phrase there? Okay. Okay. And then we go to section 11. What are the conditions under which this is accurate or not? Oh, okay. I see some real data there. All right. I see some thresholds. So, okay, I'm going to be less concerned. And those steps I'm taking calm my amygdala down and et cetera, et cetera. So if you just stop at the step one, skull and crossbones, amygdala, activates the sympathetic nervous system and you got the startle response and then your cortisol level goes up. That's great if you're in an immediate threat, like in the wild or there's a lion or tiger in front of you. Unfortunately, we don't live in that kind of situation, most of us. So it's key to go beyond that and to think about those things. So yes, there is a psychology, in my view, there is a psychology and a philosophy of both toxicity and risk built in to the GHS, which then the SDS is built on. And I think it's intentional. I think, the GHS there's a reason why it's structured and the reason why it's hazard-based. But it's unhelpful if people uncritically just use don't use all the information that's in an SDS. Let me push that.
Sure. Well, that begs the question. One of the requirements with the compliance to the standard is proper training, right? In the standard, in communication, how to read an SDS, all of that. Maybe let's have you on for a future episode and maybe we could talk about improving the education piece, the training with this in mind, right? The threat perception. Would you have some ideas for that as a future topic?
Sure.
Maybe?
Yeah, definitely. Yeah, I definitely have some ideas and,
Okay.
to even go into the literature to, on the psych side to.
Yeah, I'd love to hear that. I think maybe our audience would too. So, we'll hear some feedback certainly from us, but I'd love to, let's talk about that sometime soon. So
okay, that'd be great. Yeah.
Yeah, I love that because we don't get enough of that. The training piece is always, again, we go back to a rote thing. I got to get the checkbox cleared every year, my training for this, or when a new chemical comes into the workplace, all that. But why are we repeating the same stuff? We're going to lose that in our perception. So yeah, let's have you on for that.
Sure.
And
sounds great.
Yeah, and great discussion so far. I think let's talk about what we can give as a final takeaway to our audience. Where do you see toxicology contributing in the future to SDS creation? All of the things we've spoken about. Any takeaways for that?
Yeah, so I would say that because the assessment of hazard and toxicity is so important to SDSs that toxicology and toxicologists, primarily good toxicologists, so toxicologists who understand difference between hazard and risk, and even the fact that hazard itself or toxicity is also a function of exposure. It's not just risk. That's another piece that GHS, I think, maybe improve upon. But yeah, I think because toxicity is so important and foundational to GHS, toxicologists have an opportunity, and I think, also a responsibility to communicate, first of all, to make their recommendations based on data and evidence. First, that's foundational. If we call ourselves a science, toxicology, that's the foundation of any science, evidence and data. And then to communicate that evidence and data appropriately and to interpret it appropriately as well. There's an opportunity there, a responsibility and an opportunity for toxicologists to do that. And in whatever company, toxicologists are used, or, to help with SDS creation, or if there are consultants that are hired to help in that respect, that is, I think, that's the central role of a toxicologist. So in my view, it's not to tell people necessarily what they want to hear on either way, something's great or the sky is falling. No, it gets back to that, in my view, the Goldilocks. I think Goldilocks approach to tox, is interpreting, those data appropriately and being able to sift through reliable evidence, not so reliable evidence. And there are criteria to help toxicologists do that. But it's up to the toxicologist to educate him or herself about that, and then to do it. But yeah, I think in the future that's a key responsibility of toxicologists. And another one I would add to Atanu is to keep up with developments in the field. And so I'll just maybe, add, like the carcinogenicity. So science and toxicology have advanced, to a point where low dose linear assumptions about cancer are not the norm. And so educating yourself, taking CE courses on non-genotoxic carcinogenicity. There's a great 3 or 4 papers published in 2018, 2019. Listeners can look them up. It's called Chemical Carcinogenesis Revisited, 1, 2, and 3. You can learn a lot about, how chemical carcinogenesis is viewed today in those processes. Moved away from low dose linear, in many respects. So yeah, so I would say, educating yourself, keeping up on CE, looking outside of your organization, it's for information and CE. Those are, I think, two big pieces, toxicologists can do for themselves. I worked with a colleague many years ago, and he was interviewed by C-SPAN back in the 80s. And he was asked the question, what can we do about junk science? Why do we have junk science? And his response was, because you have junk scientists. And so, so to the extent that, junk scientists, junk scientists use junk science, junk toxicologists will use junk toxicology. So, just being aware of the latest thoughts, and evidence in the field is very important. That's what I would recommend for toxicologists going forward.
Great. Well, fantastic. The great advice. Again, this topic is very well regarded. I think it's needed for what we have out there. I'm glad you were on today. It's been a great discussion. For more information about Rob, please visit Hack Your HazCom, where you'll find this episode page and his contact information. I'm sure he'd be happy to answer questions and, get you information, like you said, to topics and papers and such, organizations that can help you with this. So again, thank you, Rob. It was a pleasure having you on.
Thank you, Tom. My pleasure as well. It's great to be here. Thank you.